evening, ladies and gentlemen. On behalf of the NYUAD Institute, welcome. My name is Timothy Dorr. I'm an associate professor of chemistry at NYUAD, and I have a laboratory and a research program that works at the interface of chemistry and biology with an interest in developing new chemical tools to study fundamental biological problems, many of them directed at the early stages of drug discovery, identifying targets and processes for drug discovery. It is my great pleasure to introduce tonight's NYUAD Institute lecturer, Dr. Mark Murcko. Dr. Murcko grew up in Connecticut, just like me, in a place about an hour's drive from New York City. He earned a BS in chemistry and applied math in his hometown at Fairfield University, and then moved about 20 minutes away to New Haven, where he earned a PhD in Yale working with Ken Weiberg. He started his career in drug discovery at Merck, where he helped invent dorzolamide, often known on the market as Trusopt, for the treatment of glaucoma. Mark is interested in disruptive technologies, and in 1990, the disruptive technology of the time was the radical idea that one could use the structure of a protein target and use computational methods to design drugs that would interact with that protein and mitigate the disease that it caused. Today, we do a lot of structure-based drug design, but at the time, 24 years ago, this was a very new idea. So he and a group of scientists founded the pharmaceutical company Vertex in Cambridge, Massachusetts, to pursue this idea of structure-based drug design. Dr. Murcko co-invented aprenavir, or agenerase, or fosambrenavir, Lexiva, for the treatment of HIV. These were Vertex's first two marketed drugs, and telaprevir, or Insevic, on the market for the treatment of some forms of hepatitis C. Until 2011, Dr. Murcko was the Chief Technology Officer and Chair of the Scientific Advisory Board at Vertex, where he guided the early stages of the cystic fibrosis drug Ivactifor, also known as Kalydeco, on the market. He is currently a principal at Disruptive Biomedical, where he continues to seek disruptive technologies that can transform the drug discovery process. He is also a lecturer at Northeastern University and the Massachusetts Institute of Technology, where he teaches courses on organic chemistry and drug discovery and mentors young scientists. In fact, it was Mark's expertise and love of mentoring that was the impetus for bringing him to Abu Dhabi, and he has spent the last 2 days talking with graduate students, postdocs, and faculty about career planning and mentoring, in addition to science and research at NYUAD. We've all been impressed by his depth of knowledge, clarity of thought, and ability to address planning issues for both short and long-term scenarios. Dr. Murcko serves on the scientific advisory boards of Syros Pharmaceuticals, Ensemble Therapeutics, and Schrödinger, among others, and on the editorial boards of many scientific publications, including the Journal of Medicinal Chemistry and Drug Discovery Today. He has co-authored over 85 scientific papers and is the co-inventor on more than 40 issued patents. He was the chair of the 2008 American Chemical Society Medicinal Chemistry meeting and the 2013 Gordon Research Conference on Medicinal Chemistry. Mark Murcko is an innovator in the pharmaceutical and biotechnology industries, an expert drug hunter, and a valued mentor to many scientists. I am pleased to welcome to the stage to present his lecture, Encouraging Innovation, How to Accelerate the Discovery of New Medicines.
Thank you, Tim. I'm really very pleased to be here, and I really want to thank you all for the very warm welcome that you've given me. I've really enjoyed the last couple days of discussions and interactions with both faculty and students. It's really very nice of you to have invited me, and I've greatly enjoyed my time here. And I have to say, it's really impressive what NYU Abu Dhabi is building. I think you really should be very proud of what has been accomplished in a very short period of time. So it's really unique, I think. So you really should feel proud of that. So what I'd like to do tonight is give you my perspective on the challenges of discovering new medicines and leave plenty of time for questions. And let's begin by asking and answering, hopefully, this question: why is drug discovery so hard? And I think, as you'll quickly see, there are many reasons why it is so hard. And so I begin with this picture. This is the very northern end of the Appalachian Trail in the United States. Some of you, I'm sure, know The Appalachian Trail is more than 2,000 miles long. It starts in Georgia and ends here in the state of Maine. This is Mount Katahdin. And there's a section of the trail right along here where the trail goes for about a half a kilometer, and it is 1 meter wide. And on either side of you, you can look down and see a 250-meter drop. And on a good day, there are 50 to maybe 80 kilometer per hour crosswinds as you're walking along this very narrow stretch of trail. Now, this is called the Knife Edge Trail. Not a bad name. And I like this as a metaphor for drug discovery because when you're doing drug discovery, you never feel stable. There's always new information coming in from all angles. You never feel like you have enough information to make good decisions. Things can change at any second. So for all these reasons, I think this really is a good metaphor because things can go wrong on the Knife Edge Trail. Things can and do go wrong in drug discovery at a moment's notice. So unfortunately, the fact is that there are very few new medicines in the world each year. And most of the medicines that are produced do not revolutionize the practice of medicine. They're more incremental. And it can take 15 to 20 years for each new medicine to come into existence, and it costs well in excess of $1 billion for every one of those new drugs. And the failure rate in human clinical studies is more than 80%. So the simple fact is mostly what people like me do is fail. That's really what drug discovery is all about. So another analogy comes from a book called The Right Stuff. Some of you may know this book. It's by Tom Wolfe. There's also a movie of the same name, and it describes the lives of the test pilots in the United States back in the 1940s and 50s. Now, these were the test pilots who were flying these brand new experimental jets going faster than the sound barrier. Now, some of you may know who this is. Chuck Yeager. It's Chuck Yeager. He was the first man in October of 1947 to go faster than the sound barrier in the X-1. Now, pilots like Yeager, all through the 40s and 50s, had a 40% death rate. And these were the guys who would go up every day in these experimental planes, and 40% of them never survived. And this is only doing this for a few years. So very, very risky business. So how did those pilots describe the fragility of their lives? They had a phrase, and the phrase they used was, "It can blow at any seam." Any error in judgment, any loss of nerve, any failure, any breakage in the machinery, anything like that could kill them in an instant. And I think that, again, is a good metaphor for what we do because, frankly, we don't know what we're doing. And I'll talk more about that a little bit later. The fact is that we can fail basically for 4 different reasons that are shown here. We might simply make the wrong molecule, a compound that isn't good enough to become a drug. Or we might not be able to deliver the molecule to the body in the right concentration, to the right tissue at the right time. Or we may pick the wrong target. We may go after the wrong strategy for affecting a disease, for improving a disease. Or finally, we may have picked a good molecule and have a good way of delivering it, and we may have picked a good target, and yet we still could fail because we may run the wrong clinical trial. We may test the drug improperly in human beings. So these fundamentally are the 4 things that can happen. These are the things that go wrong. But why do we fail? These are the things that cause us to fail, but why is it that we fail? The principal reason is a lack of information. We simply don't know very much about how to make new medicines, and I'll talk about that more a little bit later. But then in addition, there are a few complicating factors that make our lives more difficult, and they're listed at the bottom of the slide. We're working on harder problems today than we used to. The bar is set higher. For example, we all expect new drugs to be very, very safe. Drugs that were approved 30 or 40 years ago would not be, in many of them, would not be approved today because the safety is not good enough. In addition, remember I said that it takes 15 or 20 years, costs $1 billion. There are thousands of people involved in drug discovery. The process, the management of the whole process is very, very complicated. And then finally, We have what some people call fear, uncertainty, and doubt. There are mergers and acquisitions and plant closings and layoffs and pressure from Wall Street. And all of those things make it harder for big companies, big drug companies, to make the right decisions. So in a nutshell, on this one slide, I think is encompassed all of the challenges at high level that we're facing. So it's gotten to the point where everyone makes fun of the pharmaceutical industry. So this is, I think, a brilliant caricature, a spoof, of a pharmaceutical advertisement. So here we have a new drug from AstraZeneca. It's called Sucrosa. It's basically a 500 milligram sugar pill, and it is occasionally effective against every imaginable disease, but it also comes with a very long list of side effects. So I think this is a really good parody of how pharmaceutical advertising is done these days. So let me try to summarize what I've told you so far. There are very few new medicines because it can blow at any seam. The existing medicines we make usually don't revolutionize the practice of medicine. It takes forever. It costs billions of dollars, and the failure rate is extremely high, and the public's perception of what we're trying to do is not very positive. So this is just a wonderful situation. It really is horrible. And so why would people like me still be doing this, given all of that? Well, the first reason is because it really is intellectually exhilarating to do drug discovery. The very act of exploring, of looking out into the unknown, really is, it's both exciting and terrifying at the same time, like all good problems should be. So I think the quote from Hamlet, and also the one further down, this is Act 1, Scene 5, of course, this is, there's more on heaven and earth ratio than is dreamt of in your philosophy. I think that's a very accurate description of what science is like these days. But as great as I think the exploration is, as much as I love that, the intellectual excitement, even better is the fact that once in a great while, despite all of our limitations, we are occasionally able to bring a new drug to market. So in 2011, Vertex got the hepatitis C drug that was mentioned. We got that drug on the market. And then in 2012, 2 years ago, it was January 31st, 2012, 2 years and 3 days ago, Vertex got this drug, Kalydeco, approved for a subset of cystic fibrosis patients. Now, it's not for every CF patient, but one thing about CF is the patients know their genotype. They know specifically what flavor of CF they have. And so for patients with a certain very well-defined type of cystic fibrosis, it's clear that this drug has a very good chance of really helping them. And so you will find out on the web blog entries from people who've been only taking the drug for a month or a few months, and they are gaining weight, they're feeling better, their lung function is improved, they can climb flights of stairs. And so clearly for those patients, this drug is revolutionary. And that, for me, is really the most important thing about drug discovery, is that once in a while, this happens. And so it's a phenomenal thing when it does. And so you can see that drug discovery is uplifting. You know, obviously, it doesn't work all the time, but when it does, in its best moments, it reminds me of the Apollo moon missions. Now, I realize that many of you are not old enough to directly remember this, but some of you are, and I am. And I remember in July of 1969 when people first landed on the moon. And I think about the Apollo moon missions, and it's very reminiscent in some ways of drug discovery. These are huge unsolved challenges. We're inventing the science as we go. Many different types of technical experts need to be working very closely together to solve problems. It's emotional, it's risky, it's important, it engages the human spirit. I think in all of those ways, I think the parallels to medicine and to biomedical research really are fair. And so that's, again, why people like me stay in the game. But I've said a few times already that the successes are rare. And so we have to address this issue of what we don't know. We have to address the ignorance. So I think the Chinese proverb is very fitting. You know, I think that what we're doing in drug discovery is we're hunting in the dark, and we don't really know what we're doing. And so it takes a long time, and sometimes the drug is just not there. There's no drug to be found. And I think in medicine, not just in drug discovery, but in the practice of medicine, you often hear the phrase flying blind, and I think that's pretty accurate. So think about what happens. So when do you go to the doctor? When you're sick. Now think about how wrong that is. We wait until we're sick, and then we go to the doctor, so we are reacting to our illness. We're not in front of the train, we're catching up to the train, right? And when we go to the doctor has very few tools with which to measure our illness. The doctor is myopic, can't see clearly. So medicine is reactive and myopic. And Paul Yock is a very well-known physician from Stanford in California, and I think what he says here is absolutely right. Doctors are making decisions on the basis of woefully incomplete information. That's the practice of medicine. And here are some other quotes. These are from Roger Perlmutter. Now, he's an MD, PhD, who has been a leader of two very large pharmaceutical organizations, Amgen and Merck. And again, I completely agree with what he said. We really don't understand enough about how the human body works. He goes on to say, it's amazing that it ever happens that we discover a new drug because we know so little about how to fix it. And he goes on to make another point, which is actually very important. Think about how when we test compounds in clinical trials, we might typically look at, let's say, 3,000 patients. Now, what happens often is we find later that there's a very rare toxicity that may only affect one person out of 10,000. And so in the clinical trial, none of the 3,000 patients in the trial exhibited that toxic side effect. There just weren't enough patients in the trial. And so Perlmutter makes the point that even after you've studied a drug in a few thousand patients, you don't have the full range of information. And so after a drug has been on the market for a while, and it's been in many, many hundreds of thousands of patients, that's when we really start to understand all the pros and cons and the details of how to use a drug. So when we're running big clinical trials, which these days can cost well in excess of $1 billion, we have no idea whether we're going to get a piece of candy at the end or this very hard rubber ball that breaks our teeth. Because by the time you get to the late stage clinical trials, you've weeded out all the molecules that probably had no chance of being successful. And everything that's left in that last stage of clinical trials looks very good. It's impossible with our current state of knowledge, or I guess you'd say ignorance, to be able to tell the winners from the losers. Because all the molecules look quite attractive at that point. And then, as Roger Perlmutter said, it's only after a drug gets on the market that we learn more about it. So here's a real-world example. So these are 5 molecules. They're all from what's called the fluoroquinolone class of antibiotics. Some of you may have heard of or maybe even taken a drug called Cipro. Some people have taken Cipro. It's been on the market for a long time. It's been used by countless millions of people. So Cipro and another drug called Floxacin, those are both approved drugs. They're in the green boxes. The 3 molecules on the bottom with the black boxes around them, those 3 molecules at the bottom were also approved, but then subsequently they got pulled off the market because there were side effect issues. And they're very different. So there's an allergic reaction, there's a cardiovascular problem, and there's liver damage. So 3 molecules with very different issues of side effects. Now, you don't have to be a chemist. I mean, all of you can see how chemically similar these molecules are, right? I mean, these structures look very much the same. So at the time these clinical trials were running, there was absolutely no way to predict which of these compounds would succeed and which ones would fail. There simply was not enough information to have been able to accurately predict beforehand. So I've been talking about ignorance, and I refer you to Stuart Firestein, who's a professor at Columbia University in New York City, and he's published very widely on the topic of ignorance in science, and you can see a book that he's written there, and he gave an excellent TED lecture, which I refer you all to, very, very good talk. And I'm quoting him here at the top, ignorance is not a criticism. Ignorance is simply an accurate statement of fact. It's a reflection of the fact that we as a scientific community do not know enough yet. And so I think an important thing to ask, of course, after remembering the Edison line, which I'm sure you've all seen before, very accurate line about how little we know. So the question is, how can we overcome this ignorance? What can we do about it? How long will it take? So I think the first thing we have to do is recognize that many of the metaphors that we use to describe science are really not very accurate. So we are not peeling back the layers of the onion. We are not putting a jigsaw puzzle together. We're not exploring merely the tip of the iceberg. The problem with all those metaphors is that they imply that science is finite. They imply that all we're doing is collecting data, and then once we've collected all the data, we can figure out how to assemble it all and get the answer. But the science is really not infinite because, as Einstein reminds us, every time you answer a question, every new insight leads to more questions. That is the nature of science. And so I think a better analogy, also used by Stuart Firestein in his TED lecture, is ripples in a pond. Every time you toss a rock, a pebble, into the pond, the ripples move out and you interfere with the ripples from the last rock that you threw in, because all the science is connected, and every new piece of data, every new insight requires that you go back and rethink what you knew before and to re-examine it in light of the new data. Everything is connected. Now, that sounds pessimistic to some people because they think, oh, gee, there's no end to this. But I think Ray Kurzweil, who's a well-known futurist and engineer, reminds us that change is not a constant. You often hear that phrase, change is a constant. Well, it's not a constant. In fact, it's accelerating. And his law of accelerating returns points out that our knowledge is growing in all areas at an exponential rate. Many of you have heard of Moore's law, about how the number of transistors on a chip double every 18 or 24 months. But the same kind of Moore's law curve is happening in every area of science. So the knowledge is growing exponentially. So I think we should take some hope from that. I think certainly during my own career, 27 years in pharmaceutical discovery, I've seen just remarkable advances in both our basic knowledge and our technology and our ways of working together. So it really is a profound change in merely 25 years. And I think it's important also to think in longer terms because, as I think Bill Gates rightly put it, the big sweeping changes are happening all the time, but they're just not happening instantly. So people always get too excited about what's coming in the next few years. But when you look back over a decade or a couple of decades, you're always amazed. The future is always much more strange and exciting and wonderful and weird than you would have predicted it could be. And so taking that longer-term view is actually very important, I think, when asking questions about how to innovate in biomedical discovery. And I think that's important because often what you will see in the popular literature really feels like science fiction. People talk about the future of medicine in all kinds of glowing terms. And so you have people talking about, for example, artificial hearts, and you have people talking about totally robotic physicians, implanted sensors in the skin detecting almost anything you want. Entirely robotic surgery is something that you hear people talk about all the time. 3D printing of organs using stem cells and tissue engineering to produce a new liver whenever you need one. I think another good example that comes up all the time is mobile sensor networks, where you're wearing devices. You're not doing anything. You're simply wearing the devices, and they're collecting data of all kinds and wirelessly transmitting that data back to your physician, where a computer algorithm assesses it all, and if there's a problem or a potential for a problem, the doctor calls you and says, you need to come in, we need to check this out. Another example people talk about often is nanorobots that go through your bloodstream looking for problems. For example, maybe circulating tumor cells from a metastasizing tumor. And of course, everybody's favorite is what some people still call the tricorder, that old thing from Star Trek. So the idea of a handheld device that you could use anywhere, where you simply take a drop of blood and put it into the instrument, and in 5 minutes it gives you a readout on thousands of analytes. And so you can detect all kinds of problems right in the field. So these are all wonderful advances that will happen in medicine. I want to be very clear about that. These things will happen, but they simply won't happen on the time scale that some people think they will. And so if you say, I want to see all of this by the year 2020, you'll probably be disappointed. Maybe a couple of these things, the first glimmers of them will exist somewhere in a laboratory in 2020. But to have them out in the general population and in widespread use around the world, no, it won't happen. But if we take a 20 or a 30 or a 50 or a 100-year perspective, absolutely, things like this will happen. So you can see, although there's a lot of people hyping these things right now, if you take a long-term view, there's really no reason to be discouraged. It's more important, I think, to try to be pragmatic about the advances in technology and to realize how long or how quickly or how long it will be before these different things occur, and to take a long-term view. And so I think in terms of centuries. So here's an example, I think a very relevant example, to go from Kitty Hawk, North Carolina, the first glider that the Wright brothers developed in 1902, and it was about a year after this that they had their first manned flight in December of 1903, to the first maiden voyage of the Airbus A380 in France in 2005. So that's a century. So think about all the advances that had to take place during that century. Material science, engineering, wind tunnels, wing design, avionics, simulation, et cetera, et cetera. It was no one thing. It was a steady stream of innovations which added up to the ability to create the Airbus A380. And now think about how they did that. They designed it all on a computer, and they built one of them. They didn't have 10 drugs in the clinic and 9 failed and 1 succeeded. They built one, and they rolled it out of the hangar, and it took off, and it flew in a straight line, and it did a nice banked turn, and it came back down and it landed, and it didn't crash. One plane, all designed on the computer, and that was despite the fact that Airbus and Boeing did the same thing, of course, with their Dreamliner, they would freely admit that there are still many details of avionics and Bernoulli's principles and turbulence that they don't fully understand. So their knowledge is not perfect knowledge, but their knowledge is good enough to enable them to pull this off, to do something like this. Now, I freely admit, of course, that the human body is far more complex than an airplane. And so today, we cannot design computers as readily as Airbus and Boeing can design their jetliners. But again, take the long view. Think in terms of 10, 20, 50, 100 years. How far will we get? And so I've never been to Abu Dhabi before, but it just occurs to me that this revolution, this biomedical revolution, is really, if I can use a baseball metaphor, it's only in the second inning. And for those of you who don't know much about baseball, baseball has 9 innings. Okay? And Boston was heavily involved in the first inning. And I'd say we're in the second inning, and Boston is still heavily involved. But by the time we get to the 4th, the 5th, the 6th inning, who around the world will be driving that innovation? There's no law of nature that says it has to be Boston. It could be Abu Dhabi. It could be Bangalore. It could be Beijing. I don't know where it'll be. And so the beauty of this is that really we're just at the very beginning of this revolution in biomedical science. And it's a great time to be thinking long-term about where we want to go. And I think that's true in Boston just as well as it's true in Abu Dhabi. So one example that I think is very well known is the concept of P4 medicine. Now, think about it. We all want to live to be 100, and we want to still be hiking and reading the New York Times every day. We want to be active mentally and physically. So how will we get there? So Leroy Hood, very well-known scientist, one of the people responsible for the Human Genome Project's success, calls it P4 medicine, and he describes it here, medicine becoming predictive, preventative, personalized, and participatory. So what does that mean? So predictive basically means that we know which of us is likely to get which disease. Okay, that's a good thing. Preventative means that we can treat the disease before we have frank symptoms. As I mentioned before, the sensors in your body inform the doctor that you should be checked because there's a risk of you coming down with some disease. Personalized means that your disease is different from your disease. Although they're both called, let's say, arthritis, at the level of the body's own network signaling and machinery, these are different diseases, and they need to be treated on an individual basis. Each patient is unique. And then finally, participatory means that we're responsible for our own health. And so we'll be playing more and more of a role in monitoring and reacting to this information and doing the best job that each of us can to help ourselves become healthier and to stay healthy. So these are fantastic goals. So how are we going to do all these things? I've described a wonderful future, and how will we actually get there? And so this may come as a surprise to some of you, but to get there, I think it's actually very important to, for a moment, put the science to one side and talk about the culture in which science is done. And I think we need to talk about culture because, believe it or not, the culture is actually more important than the science itself. And the founder of Vertex, Joshua Boger, who was my boss for many years, I think he's got it right. Success is 98% about the people doing the science and the way they work together. And another example, a well-known book published 52 years ago, The Structure of Scientific Revolutions, by Thomas Kuhn. In that book, he makes the point that culture is the constellation of values. It's the way we think in a group, and that's what determines what problems we think are important. That also determines how we'll go about solving them. And so culture has a profound influence on the way science is done. So let's drill into this just a little bit more. So how does the culture determine just how successful I think we can be? So maybe I could just begin by describing my culture. I come from Cambridge, Massachusetts, right outside of Boston, and I think Cambridge is a pretty good place to do biotech, and it's filled with people like this fellow, Tom Hughes, who I know very well, who started life in a big company and eventually moved to smaller organizations because people like Tom are impatient. People like us, we want to make new medicines and we want to do it right now, and we're not willing to put up with bureaucracy. And another very well-respected person from this environment, Deborah Dunsire, used to be in charge at Millennium, makes it very clear. She wants to come up with new drugs to treat diseases where at the moment there are no good drugs. She wants to go after those really tough problems where people really are suffering. And I think the most important thing about this slide is that these comments, these two people, this is not unusual. This is very typical of what you see in Cambridge. This is the world that I live in. This is the culture that I inhabit. Of course, it's not just in Cambridge. So I've interviewed over the years, informally of course, hundreds and hundreds of people and teams that have been successful. And I've asked them, well, what did you do? You just were very successful at putting a drug in the clinic or on the market. Why do you think you were successful? And if you really boil down those hundreds and hundreds of interviews, it comes down to these 16 points, is that teams and individuals that are successful are far more likely to exhibit these behaviors than teams and individuals that don't do so well. So it really is fairly clear. And notice that these are behaviors. None of this is about your IQ. This is all stuff that's in each of our control. We can choose how we want to live. But how can we enable these kinds of behaviors, I think, becomes the important question. And of course, science never happens in a straight line. There are zigs and zags. There are twists and turns. There are always surprises. And so researchers need to have time and freedom and support to pursue the science through all those twists and turns. And so you have to have the right environment. And that's what Peter Medawar says here. He's a brilliant scientist, was a brilliant scientist in his own right, won the Nobel Prize, but he also ran a very important research institute in the UK, in England, for nearly a decade. And I think he got it right. You have to set the right tone. You have to build the right environment, giving people the freedom that they need to be able to explore. And that enables people to collaborate. That enables people to be curious. And curiosity is also a very important part of scientific success. And so we need, I think, and I see this everywhere I go, we need the educational institutions to support creativity, to support asking good questions and enabling scientists and engineers and mathematicians and physicians to all learn from each other, to tell stories, to understand each other's work, because curiosity in turn leads to collaboration, because scientists who are excited to learn about the work of other people who are in different fields from their own, I think often end up finding that that's a great way to advance their own creativity. And creativity and imagination, absolutely essential. I agree with what John Dewey said. As far as I can tell, I can't think of a counterexample to this statement. I think all the great new science requires that sort of imaginative, creative leap forward. So if we don't have that creativity, I don't think the science can advance. And this, it's very interesting to me that this was also known in the pharmaceutical industry. 60 years ago, this fellow, Paul Janssen, was running a lab, Janssen Pharmaceuticals, in Belgium. And he is still, to this day, the most successful, most prolific drug discoverer in history. More than 50 new medicines came out of this one company in the course of his lifetime. And there's a lot of references on this slide at the bottom, but basically if you read all of the things that he wrote and that colleagues of his wrote in memory of him after he died, there's some very clear lessons that come out of this. I mean, he made sure that people worked in teams. He despised bureaucracy. He made sure that people were working on very important, very fundamental problems. He supported people to be creative, to take risks, to measure what they were doing. So again, for somebody like Paul Janssen, success is not an accident. It's a consequence of the culture that he built and the behaviors that he encouraged in all of his scientists. And you see the same things in other technical fields as well, technical fields that have nothing to do with medicine. So Bell Labs, of course, as we all know, the folks who created, among many, many other inventions, the transistor in 1947, they had many of the same guiding principles that supported their research. And this fellow at the bottom, John Gertner, wrote a brilliant book on the history of Bell Labs, came out a couple years ago. And it's very clear, if you really boil down the formula for what made Bell Labs successful, although they did not work in medical research, every one of these points is highly relevant for success in biomedical research. So when any of us are thinking about how to build some sort of as I said, a thriving new enterprise to do biomedical research, I think we can learn a lot from studying organizations like Janssen or Bell Labs. There really are some clear lessons in here. So that really is the takeaway from this part of the talk, is that we really can see that it's about behaviors and culture and encouraging the right environment, enabling the right kind of science to be done. Now, one other aspect of that that's important, remember the science never goes in a straight line, and so it's really about being adaptable. And Darwin, of course, said it right. You have to be responsive to change. And good scientists always listen to the new data. They're always data hungry. And so if new data comes in and it forces you to change your position, that's, of course, what you do. And so the ability to respond to change turns out to be critical. And so if you think about that law of accelerating returns, think about that exponential growth in knowledge, it becomes harder and harder to keep up. And so the need to be adaptable grows and grows from one generation to the next. And so scientists, even now, but especially scientists in the future, are really going to need not just the hard analytical skills that we all think of as scientific, but also the softer skills, as Bill Drayton, who's a philanthropist, really, as he said, you really need a different set of skills. You need empathy and teamwork, and you need to be able to thrive in a chaotic environment. These are the kinds of techniques that enable you to be successful in the future. There are not going to be rules because the change comes so quickly. And so the need for adaptability just grows and grows. And I think a big part of this, and this is a quote that I know is already known to many of you. I was really surprised when I saw it up on the wall of one of the buildings because it's a quote that I've used many times. Science is global, and there are brilliant scientists everywhere. And so the best scientists are networkers. The best scientists think about how to expand their horizons. And so good scientists recognize the powers of connections, of gaining new information, new knowledge, seeing the world through different eyes. Coffee shops. 1652, London, Oxford, thriving new social network came about because of coffee shops. Lloyd's of London started in a coffee shop in 1688. And I certainly don't need to tell you guys that coffee is found everywhere in the world. And it's a fantastic opportunity for sharing knowledge and insight and storytelling. That's what this fellow is doing. I mean, that's what it means to be in a network. And so these days, I mean, we all still use coffee shops, of course, but in addition, we know that the network is global. And so it really is true that information travels around the globe at the speed of light. And so a good scientist has to be connected to the entire world's scientific community. And that means not just other scientists who are doing the same exact sorts of things that they are, but in another place. That's part of it. But it's also the people doing things very different. And learning, again, as I've mentioned before, from people who are approaching problems using very different tools and techniques and experience and judgment. And the more we do that, the more we can combine global knowledge, the more we see innovation happening around the world. And the last point I'd like to make is I've noticed this many, many times in my own career that the great teams that I've worked with, they really have a sense of being part of something bigger than themselves. And some people call this cathedral thinking. But what it really is, it's a sense of mission. It's a sense of awe and wonder. Maybe it's the same as what I was saying before about the Apollo moon missions. Maybe it's really the same point all over again. The reason I like this particular phrase is because if you read about the history of cathedrals in Europe, they often took a century to build. And so someone might be working on a cathedral and would never see the end of it. But they worked their whole career on that cathedral. And sometimes halfway through the construction process, the cathedral would collapse because the engineers were pushing past their knowledge. They were trying things that hadn't been tried before. They were inventing it as they went, just like the Apollo engineers were doing. And when that happened, they didn't give up. They kind of dusted themselves off and they started rebuilding. Just as when Apollo 1 had that tragic fire and some astronauts died, they didn't cancel the Apollo program, they kept going. And so that's cathedral thinking. And I believe that that's actually essential because I've talked a few times tonight about taking a century-long view. And I think in biomedical discovery, that's really what we need to do to achieve the global benefits that I think medicine can provide. And so I see this sometimes even in drug discovery teams, and I think it's something we all need to strive for because it produces better and better results. So let me just briefly summarize what I've been trying to get across, and then be happy to discuss this as long as you want. First, I think it's very clear that we're really bad these days at making new medicines, and as I mentioned, it's primarily because of our ignorance. It's also because the process is complex, of course. Now, it's getting better gradually, that acceleration of new knowledge, and so that's a hopeful sign, and we can look forward with confidence over the long haul that we will see steady improvements in medicine. But we also have to recognize that there are these complex challenges of behavior and culture, and these afflict scientists in companies, in academia, everywhere. It really is true that these are universal challenges. And so we have to recognize that these are issues that have to be addressed head on. It's not just about the technical challenges. So now you may think, based on everything I've said so far, you may think that these are fairly pessimistic conclusions, but I'm actually not pessimistic at all because I think there are very clear implications of what we need to do in order to encourage innovation and to accelerate the creation of these new medicines. And the first, very simply, is that we have to support basic science and recognize that we have to defeat the ignorance. And that will take a while, and that requires a long-term perspective. And I think we have to ensure that our brightest scientists are willing to consider going into medicine, going into discovery. I think that's absolutely essential. I think the second point is a little more complicated, but we have to think in terms of building cultures that enable the kinds of science I've been describing, the sorts of behaviors, risk-taking, resilience, cooperation. We have to create people who are comfortable, some call them change agents, people who are comfortable thriving in that chaotic maelstrom that exists now and will get even more so in the future. People, in other words, who can adapt to that explosive sort of growth. And so we have to build cultures that enable these things to happen, and we have to train scientists for the long haul to enable them to really adopt these sorts of behaviors. So I hope I've given you a few things to think about. Very happy to talk about these things now or in the future. I'm very easy to reach. There's my email address. And once again, please believe me when I say I really have greatly enjoyed my time here. I really would like to once again thank NYU Abu Dhabi for the invitation. It's been a wonderful 2 days for me. And of course, I'd also like to thank all of you for your attention. Thank you.
One of the issues is that it takes $1 billion to develop one of these.
Yes.
I don't want to focus just too much on the financial bit, but if we just look at that number, two questions arise. First, can you just give us some insight on how it's determined where to put the funding into? Like, how does one decide what would be a good drug to look into? Need, promise,
Yeah.
profit, didn't want to say that one. And the second question on that is, what would be the biggest drain of that $1 billion? Where does that go?
I'm sorry, the biggest?
Drain. Where does that money go
Right.
in one of those projects?
Okay, so the second part is easier, so let me start with that. The biggest waste of money is the fact that in the clinic, I mentioned that the failure rate is so high, and that often happens in phase 2. And so getting a program up to the point where you can run that phase 2 clinical trial, it's very expensive. And then the phase 2 clinical trial itself is quite expensive, and the vast majority of those fail. So the single biggest expense in the whole process is caused by the very high failure rate in the clinic. And that's because our understanding of disease biology is so poor, we pick the wrong targets to go after, or we run the wrong clinical trials. So that comes back to the first part of your question, how are decisions like this made? It is driven mostly, there are of course exceptions, but it's mostly driven by the number of patients or the severity of the illness. So in Cambridge, where I live, every drug company is working intensively on cancer. A lot of companies are working on cardiovascular diseases, working on diabetes, working on obesity. A lot of people are working on neurodegenerative diseases, Parkinson's disease, multiple sclerosis. And so the medical need drives the decision making. The problem is, because we know so little, what you'll read in the paper sometimes, and it's always unfortunate when you read these things, you'll read about a drug company that simply decides to get out of a disease area. They'll say, we've decided we're no longer going to work on neurological diseases. And usually when they do that, it's not because there's no need. It's because they come to the conclusion that they simply don't know enough and that they can't make good decisions about what to do in that area. And so I think that right now that's what drives a lot of the decision making. And that's what drives a lot of the hope, is that when you find a target where you believe strongly that you have picked a target that is responsible for a disease, then that's when you see lots of drug companies all working on the same thing, because the evidence is very strong. And so it's because of that very high failure rate in the clinic caused by the lack of biological knowledge. That's what causes drug companies to all want to work on the same targets if they're very well validated targets. Yes?
Has gone out of existence? Is it because there are new modes of coming to discoveries? And then the second question, what are the major trends in finding new drugs?
I'm sorry, how do I?
What are the major trends, new trends in finding new drugs?
Okay. So again, I'll answer the second question first. So the major trends, I think, in sort of building on the last question and the last answer, is people are far more dedicated right now to thinking about disease biology at a much more early stage. And so people are looking at genetic data. People are trying to understand whether the disease models that are being used, the animal models, for example, whether those are valid. And so there's a lot of creative things now being done, including right here at NYU Abu Dhabi, thinking about, for example, using worms in some cases. You can use zebrafish, you can use fruit flies. There's all kinds of different animal models. And so people are putting a lot more emphasis now in trying to really, as best we can, understand the disease so that we can make better decisions. So that's, I think, the trend that I see more and more. And then related to that, once the biologists have discovered a target that really does look like a good one, then it's the chemist's job to really do everything that they possibly can to make a drug for that target, to treat the disease. In the past, chemists would tend to shy away from and be hesitant to work on certain classes of targets because they were very difficult to make drugs against. These days, I think people in chemistry are a little more willing to try to work on those very hard targets if the biology looks very, very strong. So I think that's the answer to your second question. Your first question about Bell Labs, I won't profess to be an expert at Bell Labs, but they had a good long run. They were probably the leading organization, high technology research organization for 60 years. And they were in a field that had changed so remarkably. The ability to design new switching networks, the ability to design new hardware had changed so radically. The things that they used to uniquely be able to do, there were technologies coming along that enabled other smaller, faster organizations to try to do the same things. So I think that's part of the answer. Again, not speaking as an expert at Bell Labs, but that's at least I think a part of the answer.
It's quite amazing how much the drug development has changed for HIV and Hep C, and we've seen the actual disease process change, and we've seen actual remissions.
Yeah.
On the other hand, there's a lot of, I guess, the slow process around antimicrobial development, and the pipeline is pretty dry.
Yes.
We don't see much development in that area. So how do you see the science going in that direction with a lot more multi-drug resistant organisms coming and really no clear, no really new antibiotics coming to the market?
Yeah, I think that's a very accurate statement. So what I see in Cambridge, Massachusetts, is I see a lot of startups focusing on antibiotics because there clearly is a medical need and because, as you said, the large drug companies have really not been working on antibiotics. And so, the old saying that nature abhors a vacuum, well, there was a little bit of a vacuum there. And so small companies are jumping in now to try to work in this space. And so my guess is that 10 years from now, things will look a lot better than they do now. Again, I wouldn't say 2 years, but I think sort of on that time scale. So I personally, for example, am involved with 2 different startup companies, both of which are completely under the radar. They have not even announced their existence yet, but they're brand new companies working on antibiotics. And that's just the 2 that I happen to know about in Cambridge, but I know of another 10 that I'm not personally working with, but I know of. And so I do see a lot of activity in the antibiotic space. So I don't want to sound too optimistic, but I think it's a little better maybe than it looks at the moment. And I also think that the larger drug companies are starting to rethink this area because they also recognize the challenges of multi-drug resistance. And so I'm starting to see a little bit of shift back into antibiotics. Not a lot of that yet, but I do see it starting to happen. So I think you've raised a very, very good point.
So you talked a lot about drug discovery, but on the other side of that, can you tell us anything about your predictions for the next 5 or 10 years related to the regulators and the approval process of drugs? Do you think that's transforming?
So I should begin by saying, well, I should have said at the very beginning, I don't really know hardly anything at all, like Thomas Edison, 1/1,000,000%. So I don't know even 1/1,000,000% of what's going on in research. I know even less about what's going on in regulatory. So everything I'm about to say, just keep that in mind. But what I do see is I see much more creativity at the FDA. I see that, especially because the small companies are trying to do things differently, and the small companies are very smart, and so they're going to the FDA with their ideas at a very early stage, before they ever even have a molecule for the clinic, because they want to have conversations. They want to discuss, could we try this? Could we try that? Would this make sense? Would you be willing to accept this data package, or do we also need to generate some other data? And so there's a lot of that kind of dialogue happening right now, which I think is helpful. Another reason that I think we can be a little bit more optimistic is a concept called N of 1 clinical trials. Now basically what that means is that each individual subject in a trial could be viewed as their own trial. The idea is that even if you're examining 100 patients in a phase 2 study, you know that each of them is slightly different. Each of them is taking a different combination of medicines. They have a different genetic background. They have different ages. Their blood chemistry is different. Lots of other changes, lots of other differences. And so there are new statistical techniques being developed right now to try to analyze the data from clinical trials in a fundamentally different way. So I do see signs of change. Now, at the same time, the FDA is also supposed to protect people. And the EMEA, it's not just the FDA, all government regulatory bodies in healthcare are supposed to protect us. And so we want them to go a little slow. We want them to be careful, but at the same time, we want them to be open to change, and I do see a lot of evidence for that.
Actually, I have 2 questions.
Okay.
The first one is, nowadays pharmacodynamics and genotyping is highly increasing. How will this affect the process of drug discovery from cost and time? And how it will affect the different countries in the world, poor and rich and so on. The other one, about pharmaceutical companies. Is there any regulations of having just the pharmaceutical companies just making a Me Too drugs, not having innovating a new molecule for certain disease, just it's copying from others companies? Is there any regulations for these companies or no? Thank you.
Sure. So by the way, I want to applaud all of you. You're really skilled at asking questions, because everybody has asked really 2 questions, but somehow managed to make it look like, so really good job. So, so the second question is, basically any drug company can try to come up with a new Me Too drug if they want to, as long as the chemical is different in a patentable sense, right? As long as the molecule is not already covered under the previous intellectual property. They can do that. However, I didn't bring the slide, but I have it. A quote from the CEO of Celgene, which is one of the companies that in the last few years has really been skyrocketing, and is a quote, he and I were sitting next to each other at a meeting in New York in November, just 3 months ago, and he basically said, no drug company these days would ever try to create a Me Too drug. It's just really stupid. It's foolish, because you can't make any money from a Me Too drug, because that's not what customers, patients, doctors, payers, that's not what anybody needs. What we need is we need revolutionary medicine. So I think in a sense it's self-regulating, because the drug companies, I think, are smart enough to realize that a Me Too drug that doesn't have any advantage it's just not worth doing because drug discovery is still going to be so expensive. So back on your first question about what will be the role of genomics, I think it was, and epigenetics and other kinds of markers. Well, I think it will be profound. So imagine 10 years from now, by then we'll have, I don't know, 1 million human genomes in 10 years. It's a guess, a big number. And so out of those 1 million people, maybe 1,000 will have rheumatoid arthritis, and maybe 1,000 will have diabetes, probably more than that. 1,000 will have psoriasis. And so we'll start to be able to tease apart what is it in the genome that increases the chances, or the epigenome, of what increases the chances of those people ending up with the disease, because there will be other people whose genetics and other markers are nearly the same, and yet they do not have psoriasis. And so it'll be that merging of the genetic and the epigenetic and the environmental and other factors. Now, will we be completely done in 10 years? Absolutely not. But in 10 years, we'll start to have glimmers. We'll start to see dimly certain examples, and that will help us to think about where to focus our drug discovery efforts, because we'll see that there are certain pathways that show up again and again, certain proteins or nodes in a signaling network that we should focus on, because they appear over and over again in patients who have different diseases. Some people call these disease nodes. There are different terms that people use for this, but there are already a couple of examples in the literature where people have done this kind of analysis and pulled out some very interesting findings. So I think that's the quick answer. These are all really good questions.
I have only one question, but prefaced by a comment. I absolutely agree with you that your comments about how what drives a lot of this research and discovery is culture. It's a way of thinking. It's a mentality. It's not just genius, simple genius, or knowledge. And as an educator, I'm very frustrated because I realize this, and I realize it's not enough for me to teach facts or even a way to do something, but it's really a matter of teaching this mindset, this mentality. Yeah. So have you ever thought about how we could propagate that, how we can teach that?
Yeah, I have. But we all have. I won't go back to the slide, but the 16 character traits, or you could go back to any of the slides, the one from Bell Labs or the one from Paul Janssen or the 16 that I listed. It almost doesn't matter. I think what I do is I try, first of all, to tell stories. I try to help young scientists understand what leads to success. So when I'm lecturing, for example, at MIT, I often will do that in my lectures. I'll try to include stories about examples, stories that highlight examples where those behaviors have had a profound effect on people. And I also think that sometimes it can also just be done by forcing people into teams. So I see this a lot, actually, at MIT, where if you take 4 or 5 students and you put them on the same problem, and you give them a very well-defined task, but then you let them go, and then you kind of check in with them every so often and just sort of poke a little bit, and suggest different ways of tackling problems, you can, through that very simple mechanism, you can start to get across some of these things. I would say, though, that by the time someone gets to college, you could argue it's almost too late, right? You could argue that really the most important thing is education, from the ground up. Resilience is a good example. I think resilience is something you can teach, in a way, by your own example every day. And resilience is a huge predictor of success. Somebody who can get knocked to the ground and they get up and they kind of dust themselves off and just go right back in and start again. There's a huge value in that ability. So it's true for all of these behaviors. They really do require constant practice, and they have to be started from a very early age.
The catch-22 for me is we have to understand the disease biology really well to design good drugs that we think will work. And if we wait to do that, we're going to wait a very long time. And I think you would agree serendipity is just a great thing that worked for medicine throughout centuries. I think we really, if we have 1 out of 5 drugs that make it, or 20%, I think what we really need to do is do even more, test even more drugs at this stage of the game, because in a way, finding drugs that work, it also helps us understand disease biology. Then the biologists go and understand how the medicine works, and it kind of a cascade.
Yeah.
So I think really, but again, there's a funding and money limit to how much trials you can run. So we really need to revolutionary thinking around the funding process. We need, if I may call it a World Bank for biomedical development and drug discovery, something along those lines. So, I don't know where the funding ideas can come from, but we need to put a lot more effort into finding more funds for the startups and new ideas to be tested.
Yeah, I agree that more funding absolutely would be very helpful. I think another sort of interesting thought experiment, if you think about the conversation we've been having about behavior. So just as a thought experiment, what if every drug company, big or small, really exemplified the kinds of behaviors that we've been talking about tonight? How much more effective would they be? Is it a 10% improvement? Is it a doubling? Is it a 10-fold improvement? It's an interesting sort of question. I'll give you my own answer is I think we could double the productivity of the entire biomedical enterprise if we could figure out how to crack that problem. That doesn't in any way mean we shouldn't also find more research money. I agree with that too. But I do think we could come at it from both perspectives. And then the last thing I would say, your point is a good one about how long it all takes. So many labs, including Professor Dora's lab, are developing better and better what are called tools for probing biology to try to learn more about the disease state. And that's a very valuable thing. And that's consistent with my answer to one of the earlier questions about what's new and what are the reasons for failure and where are people innovating. It's really to think more deeply about disease biology. And so as we do that better and better, then we pick better targets. And so the 80% failure is happening now mostly because we don't understand the biology. So as our biology understanding improves, that failure rate should drop. So again, if we look at it over sort of a decade-long perspective, I think that's what we would hope to start seeing.
In addition to the fact that our knowledge improves, our knowledge is actually complicating the problem even more, because whereas 10 years ago or 15 years ago, we used to think that we're going to find a drug for cystic fibrosis. Now it's 20 drugs.
Right.
For cancer, it's 1,500 drugs, because now we're stratifying the disease into different targets at the molecular level. So I think an approach going back to this nodes and systems biology and stepping away from this very targeted approach, but instead to a system approach is one way of kind of making the problem a little more efficient to manage.
I think in the long run, that is absolutely, I think in the long run, that's what will happen. We will have a more systemic understanding of disease. It'll take us a while to get there, but I think that in the long run, that is what's necessary, because the human body is very complicated, and just blocking one spot, one node in the signaling machinery is probably not what's actually the right thing to do or the best thing to do for treating a complex disease. It's polygenic, it's intrinsically a mess. And so thinking that you could find one drug that would solve everybody's problem with diabetes or cancer is too simplistic. But again, because I'm an optimist, I say I'm really glad that at least we now understand that. Whereas 50 years ago we didn't, because now we have a better lens through which we can look at the problem and thereby make progress more quickly than we would without that understanding. So the understanding forced us to rethink, ripples in a pond, forced us to think differently, and that changes how we do the science.